We'll now move on to the next presentation by Kumiko Yamanaka from the Institute of Advanced Biomedical Engineering and Science at Tokyo Women's Medical University. Her title is Potential Application of Microalgae in Cultured Medium for Sustainable Cultured Meat Production. Kumiko, the floor is yours. And the pointer is, there is a pointer there, but it takes, maybe not. I think this one is the pointer, yeah.
Thank you for giving me this opportunity to talk about this, my recent research. I'm Kumiko Yamanaka from Tokyo Women's Medical University, and I currently working at Professor Tatsuya Shimizu's laboratory. So today, I'm gonna talk about the potential application of microalgae in culture medium for sustainable cultured meat production. So first of all, I'd like to, let me give you a brief introduction of my project. I'm sure all of you well known about cultured meat production, so I will go over very quickly. So, myoblasts taken up from livestock are proliferated and differentiated in culture medium and to form by tissue engineering to make this cultured meat, and scaling up is essential for the commercialization, and it is estimated that up to 40 liters of cultured media are needed to 1 kilogram cultured meat production. So therefore, the gross media is the largest cost of the production. Then commonly used culture media consists of grains-based nutrient source and supplemented with FBS. So using this in large quantity for the cultured meat production raises a number of challenges. The grains production causes the high environmental load, and the grains also needed for the food or feed, which is expected to become in short supply in the future. And on the other hand, FBS is very expensive and possess animal welfare issue and lot-to-lot differences and risk of pathogen contamination. So there is worldwide interest in serum-free and serum-free media and serum substitute. So here is our idea. So to make sustainable and low-cost culture medium, we use the nutrient, we replace the nutrients with microalgae instead of grains. And FBS was substituted with cell supernatant. Because the microalgae culture reduces the carbon dioxide and required less water, and it grows in place not suitable for agriculture, and it is high productive. The cell supernatant, also called conditioned media, contains the growth factor and cytokines and etc., which is secreted by animal cells. Using this instead of FBS is animal protective and safe and cost effective. So I will describe the development of our new culture media in 2 steps. First, we created a basal medium from microalgae, and then confirm it works with the addition of FBS. And next, we cultured the growth factor circulating cell line using this basal medium from step 1, and to see it's gonna be a grain-free, serum-free growth medium. This is our goal. So in this project, we selected the Chlorella vulgaris for my research, and then Chlorella vulgaris was freeze-dried, and extracted the nutrients by hydraulic acid hydrolysis, AH for short, and ultrasonic fractionations, UF for short. In case of AH, there the three types of extraction were made according to the degree of hydrolytic acid, and in case of UF, two types of extract were made according to the concentration of chlorella in the water. So we call them the Chlorella vulgaris extract, CVE for short. So the next we examine the nutrient contents of each extraction, and for the amino acid, the US extraction yield glutamine and asparagine, which could not extracted by AH. And for the carbohydrate, the AH extracted more glucose, while UF extracted disaccharide and polysaccharide, which said to have antioxidant property. So from these findings, we selected the best extraction for each experiment, sometimes mix them together. To make basal media for bovine myoblasts, we prepare the inorganic salt solution for cell homeostasis and added to appropriate amount of CVE. And we named it Chlorella vulgaris nutrient medium, CVNM for short. Now, FBS was added to this CVNM, and then we cultured the bovine myoblast for 2 days. And then we have done proliferation assay, XTT assay, and then as you can see here, the bovine myoblast proliferate even only a few low amount of CVE, but more than appropriate amount of CVE, the proliferation is going down. So the next we have done primary using CVNM instead of DMEM, and the yield cell number was almost half, but after several passages. So we after several passages with each the media, so as you can see, the population doubling level were comparable. After 10 passages, so as you can see this picture, the differentiation efficiency was also comparable. So we tried to make a prototype using this CVNM with FBS. So we culture the bovine myoblasts in culture dishes and harvested like this. Then after that, enzyme was added to form this cultured beef using microalgae nutrients. And after cooking, it had golden brown, and we could smell delicious aroma of roasting meat. So we successfully made the basal medium from microalgae to replace the DMEM, and we could move on the next step. From our previous study, we chose the RL30 rat liver epithelial cells called RL34 cells to use as a growth factor circulating cell line. After RL34 cells culture in CVNM in serum-free condition, we collect this as a CVNM conditioned medium and add it to the bovine myoblast culture to see if they are proliferated. This is experimental design. So to create the CVNM specifically for RL34 cell survival, we need to know which CVE or what percentage of CVE are suitable for the cell survival. So RL34 cells seeded high density in DMEM with FBS, and to attach the culture dishes. After adherent, the media was changed in serum-free CVNM, and then keep it 24 hours. Then we check the viability to find the best condition of cell viability. And this good conditioned media were applied to bovine myoblast culture, and we cultured 48 hours, and then after that we checked the myoblast proliferation. This graph indicated that cell viability of RL34 cells in CVNM serum-free condition. So as you can see, this red line, 0.5 normal AH extraction mixed with UF extraction shows the highest metabolic activity. And also the 10 to 15% is good for the cell viability. And we choose this conditioned media added to bovine myoblast culture. So this graph indicated that cell proliferation of bovine myoblast. The metabolic activity of cells cultured in CVNM and CVNM with additional CVE were 6 to 8 times higher than DMEM only. So this picture also shows that bovine myoblasts were more in conditioned medium compared with the basal medium only. So lastly, we wanted to know what promotes cell proliferation. Since RL34 cells derived from rat liver epithelial cells, and also the liver is known to synthesize insulin-like growth factors. So we first measure the concentration of IGF-1 by ELISA. So although the IGF-1 contains only 50 picogram per ml, but IGF-2 contains actual very high concentration of 120 nanogram per ml in CVNM. So it's considered to contain almost the same amount of IGF-2 as 10% FBS. So we assume that IGF-2 is one of the key factors that promoted the proliferation of bovine myoblast. So this is my conclusion slide. I think I have to skip this slide. And I'd like to thank Professor Tatsuya Shimizu, and I'm happy to take your question. Thank you for your attention.
Thank you, Kumiko, for a wonderful presentation and exciting data. Time for one question. Over there. Where? Close.
Here we go. I think we lost the top, yes. Hi. Yes, it's working. Did you check also the effects on the differentiation capacity?
For the CVNM conditioned medium.
Yeah.
Yeah. Actually, I tried several times, but honestly, its differentiation rate was slightly low. But yeah, but I think we need to more nutrition, but in this moment, the CVNM is very, very low nutrient content. So I need to figure out how to contain more nutrients. So that is a problem. Yeah, we're still working on that.
Okay, thanks.
Okay, thank you.